There are some key differences between diffusion and osmosis. To begin with, in diffusion, we have a high concentration of a solute within a solvent. The high concentration of this solute slowly dissipates and moves apart through the process of diffusion. The particles move from higher concentration regions to lower ones. During osmosis, we have the solute, the solvent, but we also have a semi-permeable barrier. During osmosis, on one side of the barrier, we have a high concentration of the solute within the solvent, and then on the other side, we have a lower concentration of the solute within the solvent. During osmosis, the only thing moving is the solvent, which moves from the area of lower concentration into the area with higher concentration.
This section is based off of a hypotonic solution. A hypotonic solution is a solution that has a lower solute concentrate than another solution. The example that we will be using today is red blood cells and water. The solute concentration for the example of the red blood cell will be around 20% and the water will be around 10%. This can lead to the statement that the solute concentration of the red blood cells is greater than the water, meaning that the statement can be made that the water is hypotonic to the blood cells. As you can see in the demonstration is of lysing in the blood cell, also known when the cell swells and bursts. This only happens in animal cells. The reason why is because plant cells have a preventative tool known as the cell wall that prevents it from lysing.
We'll be discussing hypertonic solutions through osmosis. The example we'll use is a hypertonic water solution and red blood cells, where the red blood cells are 5% and the water solution is 25% solute concentration, making the water solution hypertonic to the red blood cells. When red blood cells are placed in a hypertonic solution or surrounded by a hypertonic solution, this causes water to exit the red blood cell to an area of higher concentration. So then they will crenate, where the edges become scalloped due to dehydration.
In biology, tonicity refers to the relative concentration of solids in two solutions separated by a semi-permeable membrane. When two solutions have the same tonicity, they are said to be isotonic. In an isotonic solution, the concentration of solids is the same both inside and outside of the cell. As a result, cells placed in an isotonic solution maintain their shape and volume. Isotonic refers to a solution that has the same concentration of solids as another solution, resulting in no net movement of water across the semi-permeable membrane. In the context of biology, it often refers to the osmotic pressure exerted by a solution on a cell. Imagine you have a bag of normal saline solution connected to a patient's bloodstream. Normal saline is isotonic to human blood, meaning it has the same concentration of solids as blood cells. When the saline solution is infused into the bloodstream, the blood cells encounter an environment where the concentration of solids is the same as inside the cells.